Hello, my name is Samantha Butler, and I'm the Eleanor Leslie Chair in Pioneering Brain Research in the Department of Neurobiology in the David Geffen School of Medicine at UCLA. And I've received significant funding from the NIH, CERN, Department of Defense, and many foundations. I study the development of neural circuits in the spinal cord. While people are usually familiar with the ability of the spinal cord to control movement, I'm actually interested in a less well-known function of the spinal cord, its ability to receive, integrate, and transmit sensory information to the brain. Sensory information includes pain, touch, and your secret superpower, your ability to position your body in space without thinking about it, also called proprioception. I study a number of different questions in this area, and today in this short talk, I'm going to tell you about our work developing protocols to make different classes of spinal sensory neurons from stem cells. And these in vitro derived neurons are potential cellular therapies to replace diseased or damaged spinal cord. So where do these neurons come from? Spinal sensory neurons arise in one half of the spinal cord, and the different populations are called DI1 through DI6, and they encode distinct modalities, including pain, touch, and itch. Coordinated movement is regulated by the other half of the spinal cord. Work from our group and others have defined the signals that generate and pattern these neurons, and this knowledge has been used to direct embryonic stem cells, which are essentially a clean slate that can make any cell type, towards spinal cord neurons. We have developed two protocols, Protocol 1 and Protocol 3, by precisely defining the sequence of signals needed to first generate critical intermediate cell types, and then the full complement of spinal sensory neurons, i.e. DI1 through DI6. We've characterized these neurons by assessing whether they express the known molecular markers for neuronal subtype, and we find that Protocol 3 generates DI1, DI3, and DI2 and DI3, whereas Protocol 2 generates DI4, DI5, and not shown DI6. We've also transcriptionally profiled them, which has revealed in a very satisfying way that we are making bona fide spinal sensory neurons. So first, they have the same predicted sensory functions as real spinal cord neurons, but also they are transcriptionally indistinguishable from them. And you can see this if you map them in statistical space. Our in vitro data set maps directly on top of the in vivo data set. They cannot be distinguished. We've then used this atlas to identify new markers for DIs, and we found that different DI populations express different members of analgesic and psychoactive drug-related signaling pathways. So this is a particularly exciting finding for DI4s and DI5s, because these are the neurons that integrate pain. And this suggests that these cells might be ideal drug testing platforms to identify novel analgesics, which target the spinal cord, but not the brain, thereby potentially resulting in pain relief without addictive side effects. So the transcriptomics have also shed light on the signaling pathways that control proliferation. And we've leveraged this information to devise an expansion protocol where we passage progenitors with these proliferation signals continuously switched on. And remarkably, this modification results in very long-lived progenitors. So control progenitors die very quickly after a number of passages. However, our proliferating progenitors are dramatically expanded. They survive for many generations in increased numbers. This gives us a means of enormously increasing the yield of spinal sensory neurons. To conclude, what I've showed you is that we have generated a complete atlas of in vitro derived spinal sensory neurons that are indistinguishable from endogenous neurons. We've identified in a novel finding that psychoactive and analgesic drug-related signaling pathways are present in these different populations, which make them good candidates for drug testing platforms. We've also identified a modification of our protocol that allows us to generate sensory progenitors in very large quantities, providing a means of increasing DI yield. So this brings us to where we want to go, and we would like to do this in collaboration with industry partners. We would now like to ask, Does the transplantation of these neurons restore sensory function in an injury model? So to acknowledge the people that did the work, here is the Butler lab, past and present. The blue names are the people who had significant role in this project, but I need to single out one person, Sandeep Gupta, here with the lab dog, who is the postdoc that spearheaded this project. Thank you so much.